Hello everyone, welcome. So we are talking about the life process, class 10. We have already discussed what is life process. The first process we are coming into is nutrition. So what is nutrition? Nutrition is the process of taking in food and utilizing it. So we are not only eating, we are taking in food and utilizing it. So this is what we call as what is nutrition. Okay, so some organisms use carbon dioxide and water for they use simple food materials obtained from inorganic in the form of carbon dioxide and water. So these organisms are autotrophs. Autotrophs are those who make their own food. They obtain their food right from the organic source, and others which depends on the other organisms for their food is known as heterotrophs. Okay, and now to achieve, so the organisms use biocatalysts or enzymes. Thus heterotroph survival depends directly or indirectly on autotrophs. Okay, so we said nutrition are of two types. So one is autotrophic mode of nutrition and one is heterotrophic mode of nutrition. Okay, fine. So coming to autotrophic mode of nutrition, carbon dioxide and energy requirement of the autotrophic organisms are fulfilled by photosynthesis. It's a process by which autotrophs take in substances from outside and convert them into stored form of energy. So this material is taken in the form of carbon dioxide and water, which is converted into carbohydrates. So if you see the carbon dioxide here, we have carbon dioxide and water. In presence of chlorophyll and sunlight, it forms glucose, oxygen and water. Okay, so these processes known as photosynthesis. So this process is known as photosynthesis, and the carbohydrates which are not used immediately, they will be stored in the form of starch. Okay, which serves as internal energy reserve to be used and when required by the plants. Fine. And a somewhat similar situation is seen in us, where some of the energy derived from the food we eat is stored in the form of glycogen. So we also store the energy in the form of glycogen. Okay, next. So what are the events that occurs during photosynthesis? One is absorption of light energy by chlorophyll, then conversion of light energy into chemical energy by splitting of water molecules into hydrogen and oxygen, and then reduction of carbon dioxide to carbohydrates. So these are the steps which is taken place. Need not be in the same order, but these are the three important steps that needs to be taking place. Why not in same order? For example, say deserts. Desert plants, they take up carbon dioxide at night and prepare an intermediate which will be acted upon by energy absorbed by the chlorophyll during the day. So it need not be in right order, but all this process will be followed. So herein we can see a cross section of the leaf under microscope, and this is how it looks like. So we have the leaf here, the middle part. Okay, so when I draw a leaf here, so this will be the midrib, this is the midrib, and this will be the veins that comes out. So fine, if we take a cross section over here and we view it, we will see this particular. Okay, so we have the upper layer is a waxy layer. So this layer is the waxy layer, and above, just below the waxy layer, we have upper epidermis, and at the bottom we have lower epidermis. Okay, now this particular area, this upper epidermis where it is present till the area here, this is known as Placidae mesophyll. So this is mesophyll layer, the middle layer, the mesophyll layer, and this area where there is a lot of air space, this is known as spongy mesophyll layer. Okay, so spongy. So in the Placidae mesophyll you have the chloroplast, which is a small green, it contains the green pigments, okay, which helps in this is a site of photosynthesis taking place. And here you have the air space, so that the oxygen is present over here, and here you have the guard cells. These are the guard cells from where the entry of oxygen and carbon dioxide diffusion takes place, entry and exit. So this is the diagram of a cross section of a leaf. Okay, now coming to the activity, we need to see what are the different things. The first one that we have, so it is an activity for you to show that chlorophyll is necessary for the production of starch. So, okay, so you can say take a variated leaf, that is a part which is white and a part which is green. Just mark the area of the variated leaf. So just mark the area of the variated leaf. Say this is the green portion of the leaf. So this is the green portion of the leaf. What you will do is you will dip it in boiling water. Okay, and then after that dip it in alcohol and keep it in the hot water bath. So there will be decolorization of leaf, and then go in for iodine test. You will drop a few drops of iodine and wait for some time. You will see that this portion is becoming blue black and the other is negative. So in iodine test, the positive will be giving a blue black color, whereas a negative iodine test will be giving a brown color. Okay, that's the color of the iodine. So this is how to show that chloroplast is the chlorophyll is required for photosynthesis. Okay, Next, coming to this particular diagram, this is the diagram of a stomata that you see. So now, what is a stomata? Stomata is a tiny pore which is present on the leaves. So there's a massive amount of gaseous exchange which takes place here. But it's important to note that here the exchange of gases occurs across the surface of the stems, roots, leaves as well. Since a large amount of water can also be lost through stomata, the plant closes the pores when it does not need carbon dioxide for photosynthesis. So there is an opening of the stomata. At what cases the stomata opens? If there is a low concentration of carbon dioxide inside the cell, so then the stomata opens. In case of okay, this is the case while stomata is open, and stomata gets closed if the condition of closure is high concentration of carbon dioxide inside the cell, in case of drought or any kind of stress that occurs. So all this at night again. So as there is no requirement of photosynthesis, stomata will close. So this is how the stomata opens and closes because of the guard cells which are present. Okay, so next we are coming to this experiment, this activity which is there. So it is to show that carbon dioxide is required. The chemical which dissolves carbon dioxide is potassium hydroxide, KOH. So you take bell jars, you take 2 potted plants, keep it in a dark room sometime, cover it with bell jar. Inside one you are putting a watch glass containing potassium hydroxide. You let it stay for some time, and then you take off a leaf and test for iodine. You will find the one in A will have positive for iodine and the one in B will give you negative test for iodine, because carbon dioxide is present over there, whereas no carbon dioxide is present in A, because potassium hydroxide is absorbing all the carbon dioxide. So this is all about autotrophic mode of nutrition. Now coming to heterotrophic mode of nutrition. So in heterotrophs, what happens? The cow, the lion, everyone, they depend on other organisms for food. So they are heterotrophic modes of nutrition. There's a range of strategies by which food is taken in and used by organisms. Some organisms break down the food material outside the body and then absorb it. Example, fungi, bread molds, yeast, etc. Others will take in and then break it down within, like human beings. Okay, so there are different modes of nutrition in this case. In the heterotrophic modes of nutrition, it can be classified again into 3. Okay, 1, we have the parasitic mode of nutrition, wherein the parasite which will live on the host and harm the host. So that is the parasitic mode of nutrition. Saprophytic mode of nutrition, wherein the organism will feed on dead and decaying material, which is a saprophytic mode of nutrition that is there. Okay, and next, how is the nutrition which is being done in amoeba? In amoeba, we have the food particle which is there. So amoeba takes in the food using the temporary finger-like extensions of the cell surface, which fuse the food particles forming the food vacuoles. Okay, so it will form the food vacuoles. Inside the food vacuole, the complex substance are broken down into simpler ones. When they diffuse into the cytoplasm, the remaining undigested material is moved to the surface of the cell and it is thrown out. In paramecium, which is again an unicellular organism, the cell has definite shape and the food has a specific spot. So in a paramecium, the food enters through a specific spot. Okay, the cell has definite shape and it has had a specific spot, and the food is moved to the spot by the movement of cilia, which cover the entire surface of the cell. Next, coming to nutrition in human beings. So when coming to nutrition in human beings, we can see we eat various types of food which will pass through the same digestive tract. So we eat the food via our mouth, where we have teeth and the tongue. So teeth will break the food into smaller parts, and the saliva which is coming out of the tongue when we see something very delicious or something, there's watering of the mouth. Okay, so this saliva that is secreted from the salivary glands, this will soften the food, and also the salivary amylase, which is an enzyme which is present in the salivary glands. Okay, so salivary amylase will be secreted, which will break down the starch into simple sugars. Then the food is mixed through with the saliva and it is moved into the esophagus. So inside the esophagus there is a peristaltic movement. Peristaltic movement is the food item is here. So this is the esophagus it's moving through, and it will be a contraction. They will just contract the food and it will reach, and thus there will be a movement of the food by contraction process. Okay, so as you take out the paste from the tube, fine. Then from the esophagus it moves, the food is taken into the stomach. Okay, the stomach has large number of organs which expands when the food enters. The muscular walls of the stomach linings mixes with the food through many digestive enzymes or digestive juices, and the digestion in the stomach is taken care by gastric glands, and these gastric glands are present in the inner lining of the stomach, which secrete hydrochloric acid, then pepsin and mucus. Okay, so these are being secreted. HCl will help to maintain the pH as acidic in the acidic medium, because pepsin acts in the acidic medium, and the mucus will protect the lining of the stomach from the action of the HCl, that is the acid. Okay, so this is the thing that occurs, and pepsin will convert, it will convert proteins into peptides. Okay, the smaller, to break the proteins into smaller parts. Then after that, from the stomach, the food will reach into the small intestine through the duodenum, and herein we have the pancreatic juices. Okay, here we have the biles which is being secreted. Biles and bile dust will be secreted. Okay, so it will be regulated by the sphincter muscles, which releases it in small amounts in the small intestine. From the stomach, the food now enters into the small intestine. This is the longest alimentary canal which is filtered into the compact space because of the extensive coiling. The length of the small intestine differs in various organisms. Okay, so the herbivores, which has smaller intestine, whereas the herbivores eating grass need a longer intestine to allow the cellulose to get digested, whereas carnivores have a smaller or shorter intestine. So in the intestine, what happens? There's a digestion of carbohydrate, proteins and fats which is taking place. So it receives the secretion from the liver and pancreas for this purpose. The food is coming from the stomach in acidic and has to be alkaline because of the pancreatic enzymes to act on. So bile juice, what it will do, it will make the food into the alkaline. Okay, so fats are present in the intestine in the form of large globules, which makes it difficult for the enzymes to act on. So biles will break down them into smaller globules, and this is what is called as emulsification or emulsifying action of fats. Okay, now what happens? The pancreas secrete pancreatic juices which has trypsin and lipase. So it has trypsin and lipase. Okay, so these are the two enzymes that is present in the pancreatic juice. So here in the walls of the small intestine contains glands which secrete intestinal juice. The enzymes present in it finally convert the proteins to amino acids. So trypsin will convert the proteins to amino acids, and lipases will convert the fats, okay, to the fatty acids and glycerols. Okay, it will convert the fatty acids to glycerols. Okay, amino acids will be building up the proteins, whatever it is required in a later point of time. So in the inner lining of the small intestine, there are finger-like projections called as villi. So something like this, where we have the capillaries, and there is a diffusion of food that with the blood. So the blood absorbs whatever the food that is there. Okay. And whatever is the waste will leave the intestine and reach into the large intestine. Here also there are villi which will absorb water, and the rest which is sent to the rectum for storage and is excreted out through the anus. So these excretion or exit of waste material is regulated by anal sphincter. Okay, thank you.